Hello everyone, my name is Yash Agarwal. I am a second year MBBS student from NDMC Medical College, New Delhi. In this video, I'll be talking about transcription in prokaryotes, transcription in eukaryotes, and some other topics. These topics are very much important for your NEET as well as your board examination. I'll be talking from NCERT point of view. Plus, I'll be telling you some extra edge topics that will help you through your NEET examinations. So without wasting time, let's move forward. What is transcription? Or before that, we should discuss central dogma. Central dogma was a concept given by Francis Crick. He said DNA is a thing that stores the genetic information. And that genetic information is used to form protein. But DNA directly doesn't form protein. DNA is first converted into RNA, and RNA is used as a template to form proteins. So the first process in which the information that is stored in the DNA is copied into RNA is known as transcription. And using RNA as a template to form proteins, this process is known as translation. So what is transcription? Transcription is a process of copying genetic information from one strand of DNA into RNA. What is one thing that you should focus upon? Here, one strand of DNA. What is the meaning of one strand of DNA? Why not both the strands of DNA should be transcribed? The logic behind it is, if we transcribe, for example, this is a structural gene that needs to be transcribed. If I make a copy of it, like for example ATTGC, this is a sequence of DNA. Complementary to it, it will be TAACG. This strand will form what RNA? UAACG. And it will form what? AUUGC. This will form another protein, and this will form another protein. So the whole process will form two proteins. And you know that proteins are the one which produces physiological changes in the body. So they will have another effects. And this will be very much, it will be creating havoc in the body, right? So only one strand of DNA should be copied. Second reason, these strands will be complementary to each other also, right? So they will form double-stranded RNA, and this double-stranded RNA cannot be translated, right? This double-stranded RNA cannot be translated. So this is all about transcription, the definition of transcription. One of the most important thing is which principle governs the process of transcription. It's the principle of complementarity. You should not forget about principle of complementarity. What is principle of complementarity? The principle of complementarity states that adenine will form double hydrogen bonds with thymidine, and guanosine will form triple hydrogen bond with cytosine, right? In transcription, RNA is formed. In RNA, adenine forms double bond with U, right? Uracil. U for uracil. Now I have two questions for you. If you know the answer, comment it down in the comment box. Can DNA make copy of itself? What is the name of the process? And can RNA be converted into DNA? If yes, what is the name of the process? And name the enzyme involved in this process. So now let's talk about transcription unit. What is transcription unit? It is that part of DNA that is taking part in transcription. It includes promoter region, structural gene, termination region. Structural gene is the region that needs to be transcribed. For example, in this instance, this region is the region which I am transcribing. So it is flanked on both sides by two regions, but I will not name any one of them as promoter or termination region. There is a concept behind it. Now let's suppose I have to transcribe this strand. So I will name this as a template strand. What is template strand? Template strand is a strand that is actually transcribed. I'll use this as a template. So what do you think? My promoter region should be present on 3' end, on the 5' end of this template strand. Think. How to name promoter or termination region? You should remember that RNA polymerizes in 5' to 3' direction, right? If RNA polymerase binds to this side, then only it will be able to transcribe in 5' to 3' fashion, right? So this is your promoter region. And this is your termination region. So RNA polymerase will produce RNA that will have adenine. Now, also we don't have thymidine, so we will have uracil instead of thymidine. So this will be the mRNA that will be produced. Now let's take another instance. With the same case scenario, I had the same structural gene that needs to be transcribed. But now I have this strand, a strand 5' to 3' end. This strand to be template, I need to transcribe this. So this will be named as template strand. Now RNA polymerase will bind to which region between these two? So this will become the promoter region. And this will become the termination region. And what will the sequence of RNA transcribed? So it will start from here. This will be 5' to 3'. In this direction it will move. So G will face C, C, G, A, U, G, C, U, A, T. So this will be the sequence. You can very well appreciate that these are also complementary to each other. So if both the strands are transcribed, they will form double-stranded RNA, and that cannot be translated. So this is the reason why both the strands are not. Now, You can very well appreciate that the sequence in both of these instances matches the non-template strand, right? For example, here, if we exclude the T part, this is exactly the same copy of this strand. So therefore, the non-template strand is also called coding strand, right? Now you understand why it is called coding strand, because the sequence that is present on the coding strand is exactly the same sequence that is present on the mRNA strand. You can very well appreciate here too. See, this sequence matches this sequence. So this is the coding strand. So you know how to name the coding and the non-coding, and so the template strand will be known as non-coding strand. So now you know how to name a non-coding strand and a coding strand. So I have one exercise for you. For example, in this part, if this is the structural gene that needs to be transcribed, and I have named this strand to be template strand. So can you name what is A, what is B, and what is the sequence of mRNA? And also the coding strand. Now let's talk about transcription in prokaryotes. Transcription in prokaryotes is relatively simpler than transcription in eukaryotes. Here only one RNA polymerase is required. It is DNA dependent RNA polymerase. It will have again the 5' to 3' polymerase activity. Now, what are the steps in transcription of prokaryotes? It will have 3 steps: initiation, elongation, and termination. What is the meaning of initiation? The RNA polymerase will bind to the promoter region. Then from promoter region, it will start transcribing the structural gene, and then that process is known as elongation, and then ultimately it will terminate itself, right? These are the 3 main steps: initiation, elongation, and termination. Now explaining through the diagram. See, RNA polymerase will first recognize the promoter region, right? Now promoter region is special in the prokaryotes. It has certain sequence. It has this specific sequence: TA, T, AA, T. And this is from 5' to 3'. This sequence is known as Prinbo box. So RNA polymerase recognize Prinbo box sequence, right? Now, when RNA polymerase recognizes this sequence, it cannot initiate elongation process. Why? Because it needs a factor known as sigma factor. When sigma factor bind to RNA polymerase, then only elongation can be initiated, right? Now the RNA polymerase has started the elongation process, and the sigma factor will dissociate from it when the elongation is occurring. Now termination region will also have a certain sequence Now, RNA polymerase recognizes that sequence, but it alone cannot terminate the process. It requires another factor, that is the rho factor. So, there are two important factors, sigma factor and a rho factor, that is required for transcription in prokaryotes to occur. So let's recall what we learned. RNA polymerase is the only enzyme that participates in transcription in prokaryotes. RNA polymerase recognizes certain sequence on the DNA on the promoter region and binds to it. That region, that sequence is known as Prinbo box, right? With the sequence TAAT. But it cannot initiate the transcription process. It requires another factor known as sigma factor. When sigma factor binds to it, the transcription occurs and the mRNA forms. When mRNA starts forming, that process is known as elongation, and during this process the sigma factor dissociates from the RNA polymerase. Now, when the RNA polymerase reaches the termination region, it will again recognize some sequence, but it cannot terminate the process. It requires another factor known as rho factor. Rho factor will help to terminate the process. Now, there is some clinical correlate with this thing. Now, we know that this process occurs in bacteria, right? Do you know one antibiotic, rifampicin? This is an antibiotic. It is used for curing tuberculosis, right? How does it act? It binds to RNA polymerase, right? If it binds to RNA polymerase in bacteria, transcription cannot occur. If transcription cannot occur, it indirectly inhibits what? Protein synthesis. And we know that proteins are the things that causes physiological changes in the body. Now let's talk about transcription in eukaryotes. Before that, we should know about cistron. What is cistron? It can be similar to structural gene that we talked about in transcription unit. It is that segment of DNA coding for protein or polypeptide, right? Now, in general, in eukaryotes, we have monocistronic gene, and in prokaryotes, we have polycistronic. What is the meaning of that? It means if this is a structural gene in prokaryote, this can code for 3, 4, 5 or different number of polypeptides. But if this is the gene of eukaryote, it will only code for one protein or a peptide, right? Now, another feature that distinguishes the transcription in prokaryotes and eukaryote is that there are different types of RNA polymerases in eukaryotes. Here there are 3 different types of RNA polymerases, and they transcribe different types of RNA. The main enzyme that we need to focus here is the RNA polymerase 2, that is synthesizing our main mRNA or hnRNA. Rest you have to mug it up. Talking about the process of transcription in eukaryotes, it will have the same process. It will involve initiation, it will have elongation, it will have termination, and finally an RNA is produced. But this RNA is not mature. So we will name it HNRNA. It is heterogeneous RNA. Right? What's so special? What makes it different from the prokaryotic RNA? We know that in eukaryotes it's a monocystronic gene, but that monocystronic gene is splitted into small parts. For example, this is when these three will combine, then only it will give a particular protein. So in eukaryotes, we have splitted monocystronic gene, and this type of arrangement of gene is known as split gene arrangement. So in eukaryotes, we have split gene arrangement. What is split gene arrangement? That the monocystronic structural gene that is present on mRNA is splitted. It is gapped by certain sequences, and these sequences are known as introns. And these split genes are known as exons. So what is our aim? We have to remove these introns and we have to unite these exons to form a single monocystronic gene. So what is our aim? Our aim is to remove introns first, and we have to unite exons to form monocystronic gene. Right? Now another aim. We know that our nucleus has RNAs. So if our nucleus has RNA, we have to protect this HNRNA from RNAs, right? Otherwise it will degrade it. So how do we do it? So we have some post-transcriptional modification. See, transcription is done. The process of transcription is done. Now the next step is translation. But before that and after transcription, we have one more process known as post-transcriptional modifications, in which we prepare the transcribed RNA, to be translated into a particular protein, right? So we have three important steps in post-transcriptional modifications. First is capping, another is tailing, and finally the last step is splicing. So we will understand each and every process, right? What happens in capping? In capping, on 5' end, methyl guanosine is added. On 5' end, 5-methyl guanosine is added. And in tailing, what happens? We have adenosine residues which are being added on 3' end. So on 3' end we have adenosine residues, and this process is known as polyadenylation. So let's talk about splicing. So imagine this blue one is your DNA, or your RNA, and these magenta one are your exons. Now our aim is to remove these introns and unite these exons in order to produce a monocystronic gene. So our DNA will form snRNA, small nuclear RNA, from what enzyme? RNA polymerase 3. Right? Here we will require RNA polymerase 3, and these small nuclear RNA will form small nuclear ribonucleoprotein particles. What is the full form? Small nuclear ribonucleoprotein particles. These are abbreviated as U1, U2, U3, U4, U5. Now, what these will do? These will go and bind to the introns. U1, U2 will come, U3, U4. They will come and form a bulge here. Here also. These introns plus these SNRPs, or also known as SNRPs. So these SNRP plus the introns forms a spliceosome. But it is known as spliceosome. And then finally what happens is that this spliceosome gets broken off and these exons are united, and then this can go for translation. This can go for translation. This is very much important. So if we see the diagram which is given in your NCERT, so here is the capping done. These are the introns which need to be removed. The tailing is done. These are the splicing. These bulging part are your spliceosomes, which is formed of intron and small nuclear ribonucleoprotein particles. These are also known as SNRPs. Right? You should know this. So finally we have ended our transcription in eukaryotes. So revising once again, all the processes, initial processes like initiation, elongation, termination are same in eukaryotes. What differs is the post-transcriptional modification. In post-transcriptional modification, we have capping on 5' end by 5' methyl guanosine. And what is the meaning of this triple P? Here it means that methyl guanosine triphosphate is used as substrate, right? And polyadenylation is done by using adenosine residues. Then spliceosomes are formed using introns and SNRPs, and then finally we get this mature RNA, which is ready for translation, right? So this covers our transcription. Hope you like the video. If you like the video, click on the thumbs up button, subscribe the channel, and share as much as possible with your friends. I'll be coming back with more new videos on genetics and other topic related to NEET. So keep on following my channel. Thank you.